Hello everyone, and welcome to this my trial presentation. This is actually a test presentation that I decided to do to basically test my platform, which I hope to launch some lecture series about some science topics such as in biology, chemistry, math, and physics. Mostly biology and chemistry, some math, and so on and so forth. Right now, the platforms I think is pretty simple. I'm just using the program Cam Studio. It's a free program that you could download online, and this is the program that I'm using to basically create the video, capture the screen image, and also to record audio. I've used I think it's a program called Prism. It's a very good program that I tried, and I have tried many of them to compress the AVI files and also to change the format maybe to an MP4 or something like that to upload it to YouTube. And in terms of the background, to say the board is, I'm using the Paint program, Microsoft Paint, and so basically that's about it. As much as for the platform, I am using the mouse and the keyboard, but I think probably at some point I have to get rid of these guys because especially for the keyboard, that's very close to the microphone that Cam Studio program is using to record audio. So you could hear the chick-chack and all the noise of the keyboard when I'm typing much more than the mouse. If I click on the if I click on the mouse, the keyboard is much worse. So yeah, that's about it for that. And the topic that I have chose to to discuss today, it has to do with genetics, and basically it has to do with a section of genetics. So let me just put something down here. And so in genetics, usually different modes of inheritance and different types of inheritance are kind of classified into two sections. So you either have Mendelian genetics or you have non-Mendelian genetics. And so, okay, well, so what's the big deal with this guys here? Which one of them gets what and where? And so basically there's Mendelian genetics, there's non-Mendelian genetics. And so with Mendelian genetics there's something, it's called complete dominance. And with let me see if I can move this aside a little bit. Okay, so with Mendelian genetics you usually get complete dominance, where let's just say you have something that you got like this big A, small a, big B, and small b. And so for the individual that has this genotype, the phenotype is big A, big B, whatever those are for the genes. So these, the small a and the small b are recessive, and the only time that you would actually get an individual with a phenotype that's AB is when this is small a, and small b, and small b. Okay, so that's what that is. So you get a complete dominance, and the rate, and you get a certain ratios. Now I'll get to that in a minute. Now let me give you some examples of non-Mendelian genetics. These are genetics that were discovered after Mendel's work, which would basically be after 1900s. Now, Mendel, published, I think, his paper somewhere in 1866. Okay, check on the date, but I'm pretty sure it was 1866. This stuff I'm just going on memory. I haven't really created anything. I'm not going based on notes or anything like that, so I'm kind of going on top of my head. That's why the video might take a little bit longer, and I'm doing a lot of umming and ooming because I'm thinking about the stuff. But basically, yeah, so that I could give you some examples of Mendelian genetics. So one of the examples would be like, let's just say sex limited genes. One example of a sex limited gene would be like, let's just say the gene for facial hair. Women have it, men have it, but women, they get this fine little tiny hairs on their face, and for men it's like really tough, and the hair really builds up there. And so women might have the gene, but it just doesn't get expressed. It doesn't manifest itself, the product of those genes. So of course what's that gonna do? That's gonna change these ratios. So basically that's one example there. Another example could be something like pleiotropy. Okay, so pleiotropy, I think that's how you spelled tropy. I think that's how you spell it. And in pleiotropy, basically you have one gene that's responsible for basically a variety of traits, and so you can also see that how that can affect perhaps that can affect genotypic and phenotypic ratios. So that's something that's also non-Mendelian genetics. Another example would be polygenic traits. Polygenic traits, basically there's one trait that it's influenced by more than one allele at a different loci, which basically means there are basically more than one gene that is involved in the production of or the characteristics of that trait. That they're the alleles at different loci. Again, examples of those are let's just say human height or the eye color. Another one, you got regulatory genes. Let's just say you could have something like epistasis. Epistasis would be a good example of a regulatory genes, where basically you could have like these rabbits that have like a black and brown coat, and black big B or big B small b is dominant, and little b are recessive, and that's when you get the brown coat. Now they could also have another gene where the small a, the recessive of that gene, the homozygous recessive, is actually creates albinism. Now it doesn't matter what genes they have for the other color code, that code for the color of the code. If they have that recessive homozygous albinism gene, then also as well, then that's going to mask the characters of the other gene. And so epistasis is definitely a non-Mendelian genetics. That's also going to screw the ratios of the phenotypes as well. We're going to go into some other examples. Incomplete penetrance. So there's something called incomplete penetrance, and basically in incomplete penetrance you got some disease like, let's just say, like type 2 diabetes. You may have the gene for it, but if you're not severely overweight, or you're not severely stressed, or and you get enough sleep or something like that, then basically you're never gonna have type 2 diabetes, even if you have it. So what happened to the phenotypes? It's just like 0 now. So again, skews the ratios there. There's something else called lethal, basically genotypes. Lethal phenotype. Phenotype. Okay, so basically for lethal phenotypes, let's just say you'll get something that has like basically this and this genotypes are fine, but this genotype equals that. and that death usually occurs while like the fetus is barely forming. So it's kind of like an abortion. And usually those offspring, if they have either also other types of characters or other types of genes that you're following, they'll never make it. So if there was another, let's just say B gene that was attached to that, you know, if it was part of that, or you know, that individual, you're never going to see that. They never come out. So instead of getting a, you know, instead of getting one of these to two of this to one of recessive, all you end up is getting one of this out of 3 and 2 of this out of 3. So you get 1/3, 2/3. You never see the rest of that stuff over there. So that also skews the phenotypic ratios, and so on and so forth. And 7, of course, you get linked genes. Now, in this video, in this presentation, this trial presentation, I'm going to be talking about linked genes. So I'm just going to kind of make that red. Okay, now one of the things that we saw in here is that, let me see if I can move this up just a little bit more, because we might need the space for the, so one of the things that I usually see here is that if you look at, you know, what we were looking at here is that these things here follow. Oh, let me just put this in blue. Follow Mendelian ratios. When you get to here, they don't follow. Oh, well, they follow Mendelian ratios. Do not follow Mendelian ratios. So basically anything for Mendelian genetics, it follows the Mendelian ratios. And anything for non-Mendelian genetics, it usually does not follow Mendelian ratios, specifically the phenotypes, because in some of these instances the genotypes will look like the expected ratios that Mendel's hypothesis says that that's how it's supposed to be. So, you know, if based on Mendelian hypothesis and Mendelian ratios, if you're supposed to get like 1 to 1 to 1 to 1, then you'll get that. For instance, in some of these cases, you'll get them for their genotypes, but once you start getting into the phenotype, they'll start differing. Okay, well, in some cases the genotypes are also skewed. Like for instance, little phenotypes, both the genotype and the phenotype, they're all skewed. Yeah, you know, in the phenotype you don't even see one of the phenotypes. You never see the recessives, and also in the genotypes that recessive's gone. So your ratios is 1/3, 2/3. Yeah. So yeah, basically that's what it is. So, but what I'm going to be talking about in this presentation is about linked genes. So, well, let's go first and let's look at unlinked genes, and so to see what those kind of are. So I'm just going to put here unlinked genes. So what unlinked genes are is we're kind of basically going back to complete dominance. So unlinked genes, let's just say with complete dominance here. So we're just going to do a like kind of like a test cross. So here I'm going to set up a test cross here where I have an individual, and this individual is basically a, let's go back to the black color. So we'll go A, small a, big B, small b. And we're going to cross this individual with A, B, B. Now what we're getting here is that this individual here is called this is a dihybrid individual. Now the dihybrid individual is called dihybrid because it's doubly heterozygous for these genes, for gene A and for gene B. It has a capital A, small b, capital B, small b. And this individual here, this one's called test cross. So basically what we're doing in this instance is that we're doing a test cross. So this is a test cross individual, which is homozygous recessive for both genes. So what are the gametes? Well, what are the gametes that this individual can produce here? Well, look at this. The gametes that this individual can produce are, down a little bit more, big A, big B, big A, small b, small A, big B, and small b. So this is the gametes that this individual can produce. Okay, so I'm just gonna put something here. Just gonna write something here. Those are the gametes. How about the gametes for the test cross? Well, the test cross is just gonna have 4 maybe. So all the gametes that are gonna produce. Now, how do we get this? Well, one way to look at this is to basically say, okay, well, we could do a mathematical representation. Let's do one here. We'll do it for all of them right there. And so what we do is, well, first of all, let me tell you something about for something to be Mendelian genetics, it kind of has to follow the Mendelian laws, and Mendel's first law talks about segregation of these inheritance units. That's what Mendel called them. I've read his original paper. I've never seen anything about a microscope in there. Well, at least not in the translated one, unless it got so lost in there somewhere. He never even used a microscope. The characters that he was looking at were pretty big characters, and you could see with naked eye. He looked at, like, for instance, plant height, corolla color, pod sizes and colors, and different shapes of things like that. The seeds, the color of the seeds, green, yellow, were they smooth, were they rough, and things of that sort. So Mendel didn't even know if the genes, the chromosomes, and whatever existed or not. So he said that, well, all these units of inheritance, basically, you get one from each parent. They are conserved. They don't get mixed, as the theory before Mendel, kind of suggested, which he had to basically reject, in place of his hypothesis. And so, that these units segregate, and also these units of inheritance also independently sort. So in order to do that, we could show this mathematically. Well, first we'll do it for this dihybrid individual here. Okay, let's segregate them by positive sign. Then we could go like that, and here you could basically kind of go from there. And here's A plus A, B plus B, and this was kind of easy because you could get. This one's kind of easy because you're gonna get 2 A's and 2B. Oops, 2B. And then you're gonna end up with having 4AB. Okay, so basically, well, and what else? So what we do here is you kind of do a FOIL. It's not really, it's not that difficult there. And so you'll basically kind of go like, okay, this times this, this times that there, and then this times this guy here, and then this times that guy there. So that's how you end up with these gametes here. For these guys, well, since these are the same, you can add them. Now, I've never seen these equations. I don't think if I even seen them in Mendel's paper. I don't remember. But I think I have never for sure seen them anywhere in my schools, my book, anything of that sort, when I was going for my degree for biology. So I've never seen them anywhere. I don't know if I just kind of created it myself or what the thing is, but they do work. And these ones you could add them together, and you kind of go from there. And that's that basically. And that's where you get the gametes for this guy here. Okay, so let me just kind of go back on this guy here, and that's a mathematical representation. Now, if we did, like, for instance, a chromosomal representation, do it with this thing here. Let's see. What we could have here is we could have let's do the red, and here we have like basically this black chromosomes, and here let's just do the red one here. Okay, so here basically we have chromosome number 1, okay, and here we have chromosome number 2, and we've got this AB genes, and then here's the alleles. So we'll say, okay, well, this one is here A. Oh, oops, let me just go back to this thing here. So that one's A, and then we'll go up here somewhere and say that's B, and then we'll just kind of go this thing same here and say, okay, well, that's A, and then we'll go somewhere here and say, okay, well, that's B there. And so basically this would be the chromosome content in in the diploid cells of this individual right here. Now I could show you the sister chromatids that would form after the cell decides to enter mitosis or meiosis. We're right now interested in meiosis, or as the English would say, meiosis. So we're more interested in meiosis right now. And so basically what happens anyway is, if you wanted to really know, is that if we put this equatorial plate here, let's just say for the cell, well, that's not really a great equatorial. Well, I don't know what the heck was that, like somebody was honking the horn there. Okay, so if that's like the equatorial plane there, and so then what we could do is that, well, this represents an equatorial plane for a meiotic cell, because these chromosomes, the homologous chromosomes, are paired up. If, for instance, this was not that case, if this was a mitotic case, let me just, oh, wait, what is that color in the background? Why am I getting red there? Okay, well, I don't know why am I getting red there, but I think it's that thing there. So yeah, so basically what would happen here is that, if I only got this guy's here, and if this was a mitotic case, then what would happen is that if this is the that's not good. If that's the equatorial plane, then what would happen in this case is that this guy would actually be here, along the plane, and so would this guy. They would be somewhere around, somewhere around there, and somewhere around here. That's how they would be if this was mitosis. They don't pair up, and so they would, these guys would get pulled up there, their other sister chromosome would get pulled down here, and you get two diploid cells, and that would be the end of the story. But we're not talking about that. We're actually talking about meiosis. So that's what it would happen here. And at some point here's the equatorial plane, and so these guys will get pulled back up there. So these guys will get pulled back to one side, and these guys will get pulled back down here, and then there will be another cellular division because the sister chromatids, the imaginary sister chromatids, are still here, so they would separate, these guys would separate, but you would end up with having gametes. You would end up having gametes that are you would end up having these gametes here, you would end up having those gametes. So these gametes are these guys here, and these gametes are these guys over here. But how about these guys there? Well, we talked about independent assortment and so on and so forth. So basically what happens is that you can imagine that instead of this being there and this being, these guys could have actually just flipped over. I don't want to flip them over here because they would start looking upside down, and this guy would start looking like a cube. But they could be like that, and if they're separated that way, and these guys were separated that way, and then there was another separation between the cells. You have to end up with 4 gametes, per germ-line cell that's creating this gamete. So you would end up with having now big A, big B here, and then that's that there, and this one's here. So you can also see in terms of chromosome and kind of follow the chromosomes there. But okay, well, what are the phenotype of the offsprings? Let's see if I could just write this down here. So what do we get here basically? What are we talking about? Phenotype of offspring. Okay, so phenotype of offspring. Let me see if I could do this thing here. So what is the phenotype of the offspring? Well, basically what we have to do is we have to cross this guy with this guy's, that guy's, and this thing over here. And so what do we end up with once we cross these gametes with that one there? Okay, well, what we end up with here is we end up with 4. Now we're only going to look at the dominant phenotypes, and interestingly enough here, it's very easy. The phenotypes of the offspring for this generation are basically going to be the same as the gametes for the this dihybrid parent. So you're going to have 4AB, you're going to have 4 B, you got 4 big A, small a, so that's going to be two small B's, so that's when you get there. You got 4 small a, and then you got a big B, and you have the small b here, and then this one of course is small a, b, so you get 4 of so you can see how they kind of are very similar to the gametes that are produced by this parent. Okay, so that's one of the things here. But then, like if you really look at this, you could divide everything by 4 there, and you get a very classical Mendelian ratio of, well, let me put this in bread, of 1 to 1 to 1 to 1. Now, so let's just say if we have here, yeah, let me see if I can move this a little bit more this way. So let's just say for a moment here, if we have, we're gonna say, what I have here is the total number of individuals. Okay, so the total number of individuals in here, we're gonna have something like basically for that are representative of this phenotypes. So we're gonna have something like basically we're gonna have 20. We're gonna have something like 25, 25, 25, 25. Let's just say we have 100 individuals, and so we'll do this down here and say, okay, well, this is like 100 down here. So out of 100 individuals, we're gonna get like 25 of this phenotype, 25 of this phenotype, 25, 25. And let's just say if this is not, if I'm just 25 is hypothetical, and that's your expected. That's what you're expecting. But let's just say this number would be 28, or then this number would be 22, and some other numbers here. This could be like 29, and this one could be like 21 or something like that. Or okay. So so these ratios are going to be like 1, 1.1, 1.2 maybe. This one could be a 0.8, 0.9, whatever, but they're going to be very close. So there's not going to be much of a significant statistical, there's not going to be much difference basically. Statistically, there's going to be no large differences between what you're expecting and what Mendel's ratios, what Mendel's hypothesis tells you based on these ratios. So this is basically all linked genes. And what we have to do is that, well, what if the genes were linked? So we look at here, we say, well, they're non-Mendelian. That means they don't follow Mendelian ratios. So basically means that we're most likely not going to see these ratios here. And so let me kind of copy this here. And so I'm just going to copy this part here. We'll just go down there. Let me just, if I could paste it here. Oh, okay, so we get it. Okay, we get it there. And so basically it's just that thing there. But okay, so one of the things that we're going to look at right now is, I could have done it here too. I don't know. Well, let's just do it here and see what happens. So basically one of the things that we'll look at is that we basically kind of get the same thing. The only thing that that's not going to be the same thing, it's this guy here. And so we'll just delete that, and we'll put here now we're talking about linked genes. But we could use the same dihybrid individual. We could do a test cross again on this dihybrid individual, and somebody did this. So basically Mendel's work, which was with the complete dominance, and you see the linked genes here. So this work was done. It was kind of put away for 40 years. Some group of, at least two, I think actually 2 or 3 botanists from different independently, I think they found Mendel's paper, collecting dust somewhere in European archives, and they kind of pulled it out and they put it to the test. They put his hypothesis to the test. They saw that, well, yeah, I think it was up to something, and so on and so forth. So they kind of repopularized Mendel's work. And so basically what happened was that after like about 10 years later or so, this was somewhere like 1910 or so, some guy by the name of Thomas on Morgan at Caltech, who was working with Drosophila melanogaster, which is like the common fruit fly, gets his hands on Mendel's paper and says, well, let's apply this, let's apply Mendel's hypothesis to now animals, since they've been already, already be done with plants. So he kind of, applies Mendel's ratios to the newly characters that they have, discovered in the flies by basically blasting these poor flies with like radiation, X-ray radiation, and so so basically that's what happens there. And so they when they do the experiments, they find a lot of this type of they find a lot of this type of ratios in this type of a dihybrid with a test cross. So they find these 1-to-1-to-1 ratios here, and everything's good there. However, there are some cases that Hunt and his lab people run into, is that, well, they're doing this dihybrid with the test cross. They're doing this, but they were obtaining ratios that were of this sort, and I'm going to put this in green. I'm going to put this in green so we could tell the difference. So the ratios that they were getting is, well, they're getting a ratio that's like this, basically. A 4-to-1-to-1 to a 4. Well, what the heck does that mean, really? Well, there's pretty statistically significant difference here, okay, almost by a factor of 4. So they had to like basically reject Mendel's hypothesis because he wasn't explaining what was happening here. And so let's just say the count here was something, again this could be a little bit different, but let's just say the count here was like 40, 10, 10, and also 40. Let's just say this is the same out of 100 individuals. So now this green, okay, so they're getting this, but they're supposed to get that, and they're supposed to get that, but they're getting this numbers here, they're getting these ratios here when you divide the 40 by 10. So the hell was going on? Or sometimes they got like 4.5 to 0.5. This could be 45, 45, and 5 and 5. But one of the things that they did discover was that, let me do this here, that this ones here and this individuals with these phenotypes, that when you kind of group these two together, they've always seemed to dominate in the number of individual offsprings. The total number of individual offsprings consisted of this combination here. Now let's see, we'll do this here. See, these combinations were usually underrepresented. So let's just say in here, 20 out of, there are only 20% of these individuals with these phenotypes here. Now, so one of the things that occurred, oh my god, I should have written it down there, but anyway, oh well, so I guess link, yeah, I guess I messed up here, but anyway, it's not a big deal. I guess we already did this here, so we don't, I suppose we don't need this guy here anymore. Okay, so we'll just delete that. Okay, so anyway, well, I guess we did the other one for the linked genes, and so this is the unlinked genes. But the ratios and all this other stuff is, it's a good thing I left it back behind. So basically, what was happening here is that they saw this, and so they said, well, okay, these guys here are basically the same phenotype as this parent over here. So the phenotype of this parent is big A, big B. The phenotype of this parent is small a, small b. Wow, that's kind of interesting because they did stand out. So I'm like, this must have been like a hair raising moment here to kind of discover this part here. And so what Morgan did, that he called these guys parental types. So these guys were called parental types. So let me go back a little bit and let me just do it in the blue. So these guys were called parental types, and these guys here were called recombinant types. Okay, and these guys were called the recombinant types. But one of the things was that, well, why is there so many of these guys here? Now, one of the things that they did was that, okay, they said, okay, you know what, these genes are not on opposite chromosomes. They're most likely on the same chromosomes. That is to say that these genes are basically linked. Now, if we take, let's draw a chromosome here. So we'll say we'll draw a chromosome here, and we'll draw this chromosome here. And so basically we got, we got like something like we got A here, and we got B there, and so we'll have like a little A here, and we'll have little B there. Okay, so this is basically what Morgan hypothesized was that happening. And when when it came time to basically for the dihybrid individual, that was the time. And when it came case for these guys to basically separate from one another, and the homologous pairs, sorry, the sister chromatids, so this one kind of went its way, this one kind of went its way, and so that's where you got the gametes that this individual produces. That's this combined with the gametes that are produced by the test cross. Now for the test cross, it wouldn't matter much of, and the other thing the other thing that was proposed was that, okay, you got this guys here produce this guys produce that, but if these genes are linked, how do you get the recombinant types? And the only way that they could explain the recombinant type was that some sort of a crossing over event was happening that was cutting, that there were enzymes and other types of processes that were cutting these homologous pairs at some specific point, and then kind of annealing or putting those pieces back together, but not exactly the way that they used to be. So basically, and so the idea kind of rose that, if you have like a crossover event here, that doesn't mean much. You have a crossover that doesn't mean much. But if you have a crossover event here, then that's basically what was happening there. And if you have a crossover event that occurs here, basically what you end up with having is you get kind of this taken off, get that part taken off, you get a B there, and that's what the crossover basically produces. And so once these chromosomes segregate, and then they kind of go in their way, and you create gametes that are here, which is that, and gametes here. Now for basically this test cross individual, this is small a, small b, that's small a, small b. Even if you have a crossover, it doesn't really matter much. It doesn't matter where the crossover is. And so basically that was their explanation. And so I think that's about it for that. And so Morgan, I think proposed a way to basically calculate the frequency that was happening here. And if you know the frequency of some, or for some organism and for a set of the genes and so on and so forth, then you can kind of work your way back and basically predict these numbers. You would no longer predict these numbers based on the Mendelian ratios. You would predict these numbers based on what's called the percent recombination frequency, which is basically going to be something of this sort over here. It's going to be the percent recombination frequency, which is going to be equal to, well, it's going to be equal to two things here. Okay, so it's going to be equal to total number of offspring over the total number of offspring. Yeah. Total number of recombinant offspring over the total number of offspring altogether. So basically, for in this situation, and so for this cross that we have here, well, what is the total number of recombinants that we have? It's actually 10. So we're gonna get here, basically it's equal to 20. That's 20 divided by 100, which is going to be equals to 20. Oh, sorry, and then over here you also have to multiply this by 100. So down here, actually, we're going to get so with this, then that number will be times 100, which is going to be equals to 20%. So our percent recombination frequency for this cross involving these genes is 20%. Now, one of Morgan's students, and one of his kind of like his apostles, one of his famous students, came up with a way to represent this percent RF in a different way. So he kind of developed the first genetic map in about 1913 or somewhere around there. And so, this became known as a linkage map. Now, it's a linkage map. It's very different from an actual physical distance map. A specific physical distance map is very different. But I'll tell you that, and then I'll tell you how the map was drawn. So basically, what we have here is we're going to call this a linkage map. So this is the linkage map of basically the cross that we did up here, and we're gonna have this one chromosome. So we'll say here's just a chromosome here, and we're gonna say, okay, well, that's A, and somewhere here that's B, and we'll also give it some black marks here, and so that's where A and G, that's where the loci for gene A and GB is, and for their alleles. And I'm going to say that this part here is 20 centimorgans. So this is where that centimorgans comes in. Now, the other thing I also wanted to mention here is that when Mendel was using the characters for his pea plant, like I said, I think they're all on separate chromosomes. But it is possible that some of the characters that he was looking at and studying when he was doing his crosses and figuring out the laws of genetics, it is possible that some of those characters actually lay on the same chromosomes, but they're 50 centimorgans or more apart. Now, when you look at these numbers here, if these numbers basically were the same as here, so when we talk about 50 centimorgans, if this is 25, that's 25, we'll get about 50 centimorgans over here. But if this is 25 and that's 25, then this must become 25 and this must become 25. So when you get 50 centimorgans or more, your ratios, once you start dividing them all, they'll get 1 to 1 to 1 to 1. So what happens there? That's why I really like linked genes that are part of non-Mendelian genetics, because for some of these things here, they can really pull off both. But for linked genes, so linked genes, depending on their distance, the relative distance from each other of these 2 linked or 3 linked genes, they can get ratios that are sometimes well, at least for the 2 linked genes, they can sometimes get ratios that will start looking like Mendelian ratios, and you would not have any idea if these genes are linked or no, unless you carry out other tests. And so that's the cool thing that I like about linked genes, and that's why I chose them right now. Because if you want to get 50 or more centimorgans here, these guys will start looking. This 4 to 1 to 1 to 4 will start looking like 1 to 1 to 1 to 1, but the genes are still linked. But it is just that they're so far apart, so they're 50 centimorgans apart, and so that means that the chances of crossing over, the frequency of recombination or crossing over, is basically gets greater. Also, the other thing is that why they do this 20%, or why they do the total recombination of springs, because the presence or the manifestation of these 2 phenotypes here occurs from a single crossover. So it's basically that one crossover that creates these 2 phenotypes. As you saw, once we cross them over and change places here, so they are a product of the same crossover. And so you take that and you do it over the total offspring, total number of offspring, you get this linkage map. So you get 20 centimorgans, or you could also get let's see here, you get 20 centimorgans, or you could also get something called the 20 LMU, and that's basically linkage map units. So 20 linkage map units or 20 centimorgans, they're all the same thing. Basically, this if I could give you an analogy, for instance, for what a linkage map would be, let's assume that for a moment that like here's your house, and here's the school, and here's like 7-Eleven or something like that. Now let's just say your friends, sometimes your friends come over from school, to your house, and sometimes they come over from 7-Eleven to your house. You've timed them, and that they don't stop along the way, and they walk at the same rate and all that stuff. Everything else is pretty much controlled for, but you see that every time, or on average, your friends who coming from 7-Eleven, they get to your place much sooner. So just from that, you can infer that, well, 7-Eleven is much closer to your house than the school is. Yeah, you don't know the distance, but you could tell that from that data that's given. Okay. But of course, when you go out there and you measure, well, you see maybe this is something like, maybe you see that this is like 1 mile, and this is something like, I don't know, 5 miles or something like that. And so that's why, so this would now be a specific physical distance that has been measured. So it's the same thing for the linkage map. For the actual map, you would have to measure the distance here by base pairs, or actually kilo base pairs. So you would have to use kilo base pairs. So there may be 2 or 3 kilo base pairs, which is kilo base pairs, 1,000 base pairs. So they're about 3,000 base pairs apart. That would be actually a physical measured distance. These are relative, so basically it's the position of this known genes, relative to each other in terms of recombination frequency. This 20% that we see here, they're basically about this 20%, this 20 centimorgans, the 20 linkage map units, they're all the same thing. Now, you know, these things really get interesting when you start throwing in 3 or 4 genes and so on and so forth. You could usually do different, you know, you could do those usually different. What you could do is you could do a cross between A and B, and then you could also, let's just say, we'll throw C over here. You know, there's C right now. And you could do, you know, so what you could do is you could do like B and C. Then you can figure out the distances. Then you can figure out the distance from A to B and A to C, and then you can figure out the distance from B to C, for instance. And this could be something else. So let's just say this would be like 2 centimorgans or something like that, and then the total from here to here would be 22 centimorgans. So it can really get interesting. Or you could just do one big tri-hybrid test cross, and then that gets really interesting. And the interesting thing about it is another interesting thing about this would be that, well, it doesn't matter if you look at it down this way or look at it down this way. If C is here, A is here, C was here, A was here, B would always be in the middle. How do you figure out that B is in the middle? You know, because you wouldn't notice to start with. So that's one interesting way to figure out. Then you can also figure out these ratios as well. So it does get very interesting with tri-hybrid crosses. It gets very complicated, but it also gets very interesting as well. Okay, well, I hope you guys have enjoyed this presentation. You know, I hope that you guys can maybe leave some comments. I was also trying to do, you know, a little bit I was gonna try to do the background in black. Okay, so basically I was just gonna kind of like, you know, make the background all black. You know, instead of the white, because if you have to look at this video for like, I don't know, 20, 50 or 1 hour or something like that, 20 minutes, you know, 50 minutes, 1 hour, I think it takes a toll on your eyes basically. So maybe I might do the videos in black, because that's what usually I've seen them do that. I've seen the masters do stuff like that. So Sal Khan usually does them in black, and so on and so forth. So I think black might be a little bit better. And I definitely think I have to get the I have to add the a stylus and also a drawing or digital drawing or writing tablet to my platform. And I will do that later. But yeah, I hope you guys can leave me some comments in terms of maybe what else I can add to my platform, which I hope to launch these type of videos in the future. And you know, any other comments or anything else, you know, something like that, like I suck at explaining this stuff or anything like that, would be nice and cool. But yeah, that's about it. Yeah, so I hope you guys have enjoyed this, and I'll basically stay tuned for further videos.